I would invite Dr. CraigSturrock to his presentation. Craig is a Principal Research Fellow at University of Nottingham School of Biosciences in the UK. He works on X-ray imaging for rhizosphere research, and he's developing methods to quantify soil-plant microbe interactions and has used that also within the TOMRES research project. Craig is the co-leader of work package 2 as well. Craig, the floor is yours.
Thanks, Hannah. Okay, good morning, everyone, and thanks for the opportunity to share some of the work that I've been involved with on TOMRES project relating to the sort of quantification of root traits in tomatoes using X-ray tomography. So as Hannah said, oh, I'm not changing slide. Oh, there we go. Yeah, as Hannah said, I'm based at the Hounsfield facility at the University of Nottingham. This is an X-ray computed tomography facility dedicated for soil and plant research. It was opened in 2014, and we've got it's kind of dedicated facility. It's got an integrated glass house, and we've got 3 CT scanners there that allowed us to sort of characterize both the soil and plant root structure non-destructively. We've got a small scanner for looking at kind of soil aggregate kind of scale interactions. The medium scale scanner is the one that we use in TOMRES, and we've also got this sort of large scale CT scanner for sort of 4 more field relevant applications where we grow the plants in the greenhouse in large 20 centimetre diameter pots that are about 1 metre length, and we've got an automated robotic sample delivery system where they're delivered into the scanner, completely automated. So yeah, within the talk today, I'm going to give you a bit of introduction into the sort of use of X-ray CT for imaging plant roots in soil, and then give a few examples of applications within the TOMRES project. I think I met at the start that my background's more towards the sort of soil science area. That's where I started using the X-ray imaging technique. So yeah, so I'll be a bit more of a sort of technical focus on the technology rather than sort of plant biology that's I'm not so strong on. So I'm going to kick off with showing this little video of some soil, and it's this is quite a high resolution image of a soil and that we've collected using the CT scanner. We can sort of slice into this soil and we can see all these sort of individual aggregate structures of the soil. And I think the reason I'm showing this just now is really to sort of highlight how complex the soil environment is for any plant growing through. So as the roots of the plant grows through this sort of heterogeneous environment, they're going to encounter different moisture levels, soil strength levels, nutrients, interactions with different biotic organisms, whether bacteria or fungi, and they're all going to contribute to how that plant responds in the natural environment. And obviously sort of going to sort of the plant phenotyping elements, obviously fairly important in this, and how the plants respond is key to how they're interacting with these local environments. So within using this technology, we can use it to sort of phenotype the roots and understand and characterize how they respond to the environmental conditions. So under these sort of drought conditions or plants that are adapted to drought and be able to sort of change their phenotype and response, can actually sort of pull out, maybe access deeper layers of water in the soil or harvest nutrients more effectively. Whoops. So using such, we don't visualize the roots in soil. There's lots of different methods to be able to do this, but it's very challenging. Soil's an opaque environment, so being able to pull out the roots is traditionally been quite difficult. Usually would involve washing the roots out from the soil. There's drawbacks of that, although it's quite not costly to do. It can sort of lose parts of that root structure that might be important for understanding how the plants interact with the environment. This shovelomics technique's been developed by Jonathan Lynch's lab in Penn State University, and it's really been quite an amazing sort of low cost solution where you're just using a spade really to dig out the sort of soil round about the root systems and characterize the roots using sort of smartphone technology and some image analysis techniques. And they're able to sort of work through quite large sort of field areas quite quickly doing this. How much higher resolution? There's techniques like thin sectioning to give you an more interaction of sort of what's going on round about the roots and the soil, but it's very laborious. You have to sort of encase your samples in resin and then do this sort of thin sectioning. It can take kind of weeks, if not months, to get some of the data. So at the University of Nottingham, we've been sort of keen on this X-ray imaging technique. It was sort of developed late noughties, kind of early 2000s when people started using it, because there was more sort of commercially available X-ray CT systems coming on the market for sort of engineering and non-destructive applications. Nottingham, we purchased, well, actually, with some of the first CT scans that we did, we actually used the local hospital CT scanners. This is part of Sasha Mooney, that's our group leader, did some of this work where you take in large pots of soil into this, the hospitals at night when nobody else was using them, and did some X-ray imaging. So you can see this sort of large soil aggregate here with some plant material going in. And then later, he got funding to sort of buy our first CT scanner, which is when I joined the university. And we can sort of see these sort of a root system that we pulled out from one of the pots. So within TOMRES, we have grown these tomatoes at quite seedling stage. So generally they're pre-germinated for about 4 days, and then they're grown in a sort of mixture of sort of sandy soil, which is much easier to sort of control the nutrient level there. We wanted to start off with something that was quite low nutrients and be able to supplement it to get a higher nutrient status. In this project, the pots are about 7 centimetre diameter, about 15 centimetres long, and the plants are grown for 14 days. And the reason it's, obviously, we're really just at an early establishment, a growth stage, because if you grow them for much longer, the roots become pot bound, and you can be able to pull out this sort of, a nice sort of 3D architecture becomes much more complicated. So once we have our plants grown and ready to be scanned, this is a picture of one of our CT scanners. So it's basically just a big lead line box to protect the operator from in the lab from any of the X-rays escaping. And then inside the scanner, there's an X-ray source. So where to put your sample, and then there's a detector panel just out the side. And we take a series of X-ray images as the sample rotates around 360 degrees. And then this particular experiment, we're actually imaging this in 3 individual sections. So there's probably about a total of about 6,000 images that are taken. And then this computer software reconstructs all this to give sort of back project a 3-dimensional volumetric data set. So this is one kind of cross-section image through that pot. So you use the software and slice through it. And then this is sort of going in through from the front hand side. So the boxes here are how that it's been sort of scanned top, middle and bottom, and then the software automatically stitches it together. So what you see in the images, all the darker areas are the lower density material, so the airspace round about the pot. And then as the contrast increases and you've got these sort of brighter little sand grains and things in those images. The big challenge really is how you go from these sort of slice images to get this sort of nice 3-dimensional root system from the pot. And it's all based on the sort of colour differences between the soil and the roots. The roots are lower density, but there's also a lot of other material in that soil, which is organic material, sort of partially filled water pores, all have quite similar contrast to the roots. So going from these images to the 3D images is really, it's still quite time consuming, and one of the main bottlenecks that by using this technique. It is possible, like we've been working with colleagues in computer science, they've had some real big breakthroughs in the last 2 years with using deep learning algorithms, where we can automatically sort of train a neural network to pick out where the roots are in comparison to the soil. And ultimately you get these sort of nice three-dimensional structure of the root, and obviously it's structures dependent on the environmental conditions around about it. So we can start to define root traits here. So we can get sort of root counts, length, total volume and surface areas, the branch structure, some of the more complex ones for understanding what's going on in the soil. You can get an idea on what the centroid values, so the weighting of where the branch structure is. Is it bushier at the top compared to the bottom? And also things like the convex hull, which gives you an idea of how the sort of volume of the soil that's actually been explored by the root system. And you can pick up sort of finer lateral root structure. Because it's non-destructive, within TOMRES we've just been using one single snapshot to sort of screen the different lines, but it is possible to do this sort of temporally, and you can actually map the sort of three-dimensional structure as it evolves over time and sort of get that temporal aspect as well. We've used that in other projects. So within TOMRES, all my work's been sort of centered around work package 2, task 2A, where we've been investigating the contribution of the root system architecture to combined abiotic stress. So the lines that we had to, obviously we could, because the technique takes quite a long time to actually get the data, we had to really sort of select a subset of lines. Obviously, in Danny's talk, he's been screening sort of thousands of these different tomato varieties using X-ray CT to do sort of phenotyping. It's a much lower throughput, so you really need to sort of use a different technique to sort of pick up some key individuals that might be of interest. So here we kind of used the data from the University of the Balearic Islands in Mallorca, where they'd done this sort of field trial, and we picked out some of the lines that were either sensitive or resistant to the combined stress, and targeted those for doing some X-ray imaging on. We started off with M82 as a control line, so that was our TOMRES collection T162. And here we've got just a well water treatment with 100% nutrients on one side and 50% on the other. And all the plants grew quite well. There's got a lot of root structure in there. We can see that there's these sort of long lateral roots in both of them. The growth response was slightly lower within the lower nutrients. But we compare that to the drought stress treatments, we've got quite a different root structure here. The lateral root developments have certainly not grown quite as wide into the soil. But interestingly, when we sort of compare out some of measure the traits, obviously the root volume is much higher in the well water condition compared to the droughts. But then when we looked at the lateral root number, although they were shorter, there was often slightly more number of lateral roots within them. So you can sort of pull out these differences between the response of the different treatments. So the project sort of went on, we just sort of screening out these sort of different lines. So we've got a sort of subset of the ones that we looked at, and sort of pulling out these sort of differences between both the combined stress treatments. So some of them certainly responded quite differently, but it's going to be quite difficult working out sort of what the sort of genetic basis and things of between these differences are, just based on these two here, the T113, 313 and 327 were probably they're both the same variety. They're a ramelet variety from the use in Mallorca, and these are, it's quite striking the difference between these. The 313 is much lower number of root biomass compared to the one that was identified as being more resilient to combined stress, where it's got much more roots. So that could be contributing to why it's sort of more resilient, certainly in the field trial data, to combined stress. We could some of these metrics go looking through. So certainly within the combined stress treatment, we often see quite a common reduction in the amount of total root length and the biomass, whereas some of the ones like 250, there is a bit more of a, certainly not a reduction, but an increase there. A lot of the variability was quite high in these, so they're more sort of trends in the data rather than significant differences. Okay, we just sort of see some of the root numbers. Again, we've got these sort of decreases, slight increase in the 250. Root angle response is one of the ones that perhaps we've seen in cereal crops under drought conditions. We get a sort of deeper shift in root angle as the roots grow down and sort of try to access water deeper into the soil profile. Again, variability was quite high across the ones that we looked at. 250 was interesting, though in the combined stress we did see more of a significant difference between these 2 points here. So there's sort of suggestions that there's things going on there. So quick kind of summary on outlook on this work. There's clearly differential responses in the root traits to the combined stress. Some interesting things that we've been thinking about is whether or not the combined stress can cause a fundamental shift in the root system trait structure, or is it just like the values are, the growth isn't quite as much, so you're under the drought conditions. Explore this a bit further. So we've done with temporal imaging, and obviously we'd be keen to explore what the molecular mechanisms behind some of these changes are. We say that we're kind of there's some limitations to the method. It would always be interesting if we could actually sort of grow these out to sort of yield, but the plant pots are really too small to restrict doing this. And still the throughput's a challenge to sort of start screening more lines. The second area that I'm going to quickly talk about in the rest of the talk is some work that Veronica Santoro, it's the University.
We've got 3 minutes.
Okay.
Craig, you know.
At the University of Turin in the soil chemistry group, Veronica visited my lab in 2019 to do some follow-up work on some interesting phenotypic differences in the work that she's been doing in her PhD. So she's been looking at the effects of strigolactones on the tomato plants under P stress, phosphate stress, as part of sort of investigating what the contribution to the root exudates were on the project. So yeah, so this is an image of some of the plants that she's been working on her PhD at Turin. So we've got a wild type, the M82, compared to this CCD7, which is the strigolactone silenced lines that Francesca mentioned in her talk. So within the silence one, it's sort of a dwarf phenotype. There's a much more sort of increased branching. So Veronica was interested. These plants have always got different phosphate uptake characteristics as well that Veronica was interested in exploring more using X-ray imaging. I'm going to sort of jump through some of these. So yeah, she visited in 2019 and did some nice X-ray imaging experiments where she grew these 2 lines in a quartz sand, so they could really sort of cut out the phosphate within the system and then supplement it for the high P conditions. And the plants were grown for 10 days and then imaged. Got these lovely images of differences between the plants. So in terms of primary root length, there wasn't really much difference. All the roots grew all the way down to the bottom of the pot. However, the difference, certainly with the lower phosphate content in the soil, the lateral root length was much shorter compared to the one with the higher P. And Yeah, so that although there wasn't significant differences between the primary root length and the angle, these true rates were kind of linked to the plant's capacity to explore deeper portions of the soil. These results weren't quite as expected. Obviously, we were looking for that sort of bushier, more branched root structure. So when Veronica went back to Turin, she did some additional work using WinRhizo. I got much stronger response, and it was possibly because our plants, when we grew them at Nottingham, were grown with the low phosphate all the way through from germination. In this experiment, when using the WinRhizo technique, they were actually sort of grown in full conditions and then sort of starved for phosphate, and it sort of switched on maybe this difference. And yeah, so Veronica got this nice work published in Plants last year, supplemented with all these sort of nice microscopy images as well that showed sort of differences in the mucilages surrounding the root trip, and also sort of like exudates on the root hair. So yeah, lovely images. So yeah, so just sort of quickly sum up. So the X-ray technique's obviously kind of non-destructive, as shown. It's got quite it's maybe got wider application to look at sort of temporal dynamics non-destructively. There's still obviously big differences, big challenges on throughput, but it does allow us to sort of capture differences in the sort of soil structure as well as the roots. And there's obviously opportunities to explore a whole range of different things in the future. So just like finish up and say thanks to the team at Nottingham. Fresh to Mansurani was the postdoc on the project. Gupin Sons was the technician that worked with a little bit. Obviously thanks to Malcolm and Graham, who's my colleagues within plant science. Team at Turin, Veronica, Lucella, and Francesca and Andrea, and all the other TOMRES partners. Thank you.
Thank you very, very much, Craig, for this good presentation that also linked up with Francesca's presentation before, really showing how the different facets of TOMRES research interact and are integrated.